So for today, the topic of study is organ of Corti. To understand organ of Corti, somebody would ask you where the organ of Corti is located. We would say it's on the basilar membrane. Here in the diagram, you can clearly see a flat base which is written as basilar membrane, and above that you have a triangle through which there is rather than a triangle, it's two rods, right, and there is a nerve fiber which is passing through. The nerve fibers are the unmyelinated nerve fibers, and that space which is below that is called as the tunnel of Corti. Importantly, as we go anteriorly to that triangle, we have the dater cells. These are super important cells which are the supporting cells of the hair cells. So before we understand that completely, let's understand the structure, the components, the mechanism of hearing, and the clinical parts. So for the structure, we spoke about the basement membrane, we spoke about the triangle as in the rods, right? And we have the nerve fibers which is passing by, that is the unmyelinated nerve fibers. Anteriorly to that we have drawn the outer hair cells, and posteriorly to that we have written the inner hair cells, right? And importantly, we have to know about the hair cells, we have to know about the dater cells or the supporting cells, then we have to know about the tectorial membrane, and we have to know about the basilar membrane. To know exactly about the structure, remembering this diagram is important. So an extension of the tectorial membrane, as we go posteriorly, we draw a Reisner's membrane. Then anteriorly to the tunnel of Coty, we have the dater cells. Anterior to that we have the cells of Claudius, we have the cells of Henson, we have the Bocher cells, right? Now if you were to draw the tectorial membrane, you are expected to draw a gelatinous matrix plus fibers, right? Now this force between the tectorial membrane and the hair cells produce the stimulus of hair cells. Like we know how sound is produced, don't we? That would be explained in further videos. So this is like the basic structure of it. Now let's look into the components of it. If you were to look about the components, first is the tunnel of Coty. This is the most important component which we talk about. So this tunnel of Coty is formed by the inner rod and the outer rod, and it is fluid filled, and it is called as Corti Lymph, right? And we do not know the exact function of the rods or the Corti Lymph. So tunnel of Coty just exists. That kind of helps us demarcate a lot of things. Then we talk about the hair cells. Well, this is very important. What are hair cells? See, hair cells are basically receptors. These are the receptors which are present in two forms, mainly the inner hair cells and the outer hair cells. Mainly these receptors or these hair cells which are the receptors, which help in two main things. One is the transduction of sound energy and conversion of sound energy into electrical energy. Well, almost similar sounding, right? Now we have two types of cells, as I told you, that is the inner hair cells and the outer hair cells. What are the differences? The inner hair cells, see, the diagram I've made are floss shaped, whereas the outer hair cells are cylindrical in shape. And I've done less number of inner hair cells compared to outer hair cells. That means outer hair cells are greater than inner hair cells. Having said that, the outer hair cells are present in rows, whereas multiple rows, like 3 to 4 rows, whereas inner hair cells are present in one row. This triangle or this two-sided figure which I've done is to represent that. Anteriorly to that we had read about outer hair cell, and posteriorly to this triangle we had seen the inner hair cell. The inner hair cells, talking about the nerve supply of the inner hair cells, we have the afferent fibers which are mainly linking into the inner hair cells, whereas the outer hair cells have afferent fiber linked to them, right? It's not exactly, it's not like specifically only afferent fibers. The nerve supply of the inner hair cells are afferent fibers plus efferent fibers, but majority of them are afferent fibers which go to the inner hair cells, which are supplied by the inner hair cells. But to understand what is all this nerve fiber, understand this. Once sound comes in, once there is compression and rarefaction, it comes through the external auditory meatus, it goes and hits the tympanic membrane, hits the ear ossicles, hits on the stapes footplate, then it goes upon to the canals, the fluid. There is this transmission of the vibrations through the fluid, which goes and vibrates the membranes. What membranes? The basilar membrane. There the hair cells are stimulated, the basilar membrane, right? the tectorial membrane. These membranes are stimulated. Once these membranes are stimulated, the air cells are stimulated. The hair cells, like the inner hair cells and outer hair cells, convert this mechanical form of energy or sound form of energy into electrical form of energy and supply it into the nerve cells or to the nerve fibers. So the inner hair cells are mainly connected with the afferent fibers, whereas the external or the outer hair cells are connected with the efferent fibers. Having said that, talking about function, the inner hair cells are mainly involved in the function of transmitting the auditory stimuli, whereas the the outer hair cells are involved in the modulation of the inner hair cell function. Having said that, so this is mostly the function of the hair cells, but importantly remember the vulnerability, the damage. So the inner hair cells are pretty strong. They are in single row, right? They're more resistant to damage, whereas outer hair cells can easily get damaged. That is one point we have to remember extra. Then supporting cells, we have already seen them. What are they? They are the Dieter cells, then they are the Hensen cells. The Dieter cells are something which support the outer hair cells specifically. We can see them just below the outer hair cells, right? And we have the Hensen cells which lie outside the Dieter cells. Then you have the tectorial membrane. Well, this is important. I told you, right? We have to remember that as a gelatinous matrix with delicate fibers. Well, this overlies the organ of Corti, and the force between the hair cells and the tectorial membrane. This force between you can see here the hair cells and the tectorial membrane. This leads to the stimulus of the hair cells. Having said that, let's quickly look into the physiology of hearing, right? See, any sound wave will have a set of compressions and rarefactions. So this frequency of how many compressions, rarefactions are there, we'll talk about the frequency, will lead to the pitch, will ultimately denote pitch. Whereas the length of this compression rarefaction will talk about amplitude, which will ultimately talk about the loudness. Having said that, we know the sound passes through the outer ear, middle ear and the inner ear, and ultimately reaches the oval window. Right? Now let's understand this. So this is the oval window, and in the oval window we can see some set of sound waves which are passing by. These are the traveling waves, right? Then you have the scala vestibuli, then you have the scala tympani, then you have the basilar membrane. So the basilar membrane is kind of parallel, like this. Now this is the cochlea, right? This early part of the early part or the open-ended part is the base of the cochlea, whereas towards the tip it's the apex of the cochlea, right? You have the scala vestibuli, then you have the scala tympani. Now what happens is, whenever there is sound wave coming in, it vibrates the basilar membrane, which stimulates the receptors, right? What receptors? The hair cells, right? We saw that on the basilar membrane only did we have the organ of Corti with the hair cells, right? So whenever sound wave comes in, it vibrates the basilar membrane, it stimulates the receptors, right? Now high frequency sounds, the tendency of the cochlea is that at the base of the cochlea, high frequency sounds are red, whereas the low frequency sounds are red at the peak of the cochlea or the apex of the cochlea. Remember this. So if you were to draw a line from the base towards the apex, you can write an arrow mark saying that increased sensitivity to low frequency. Now there is a characteristic feature about the basilar membrane. The basilar membrane, as we go ahead from the base to the apex of the cochlea, becomes thicker and thicker. It has a gradient, right? It becomes thick. The base of the cochlea of the basilar membrane, the base of the cochlea is thin and stout. As we go ahead, the base of the basilar membrane, which is at the apex, the basilar membrane at the apex is thick and loose. Having said this, now what happens is, basically whenever there is sound wave passing by, there is vibration, there is stimulation of receptors. This will go through the auditory pathway and it will go to the auditory cortex. Now what does the place theory of hearing say? We are talking about theories of hearing. What does the place theory of hearing say? Now, so any pitch which is by frequency, so any pitch encoded in a particular place in cochlea via the receptor stimulation is called as the place theory of hearing. Simply means, as sound waves passes by the base to the apex of cochlea, any place there will be stimulation of hair cells which will basically encode to the pitch of the sound, and that will simply talk about the place theory of hearing. But now there is a shortcoming of place theory. Now sound has a large range of frequency, right? That is from 20 hertz to 2000 hertz, 20,000 hertz. So 20 hertz to 20,000 hertz. So the place theory was not able to completely explain the whole process. So we needed two more theories. One was the frequency theory and the Wally theory, right? The Wally theory and the frequency theory, which spoke about the pitch encoding itself. Now, what we need to understand is again the same diagram of the cochlea, right? Now, as we look at this, right? So, what does the frequency theory say? What does the frequency theory say? It says that every sound wave, every up and down of a sound wave encodes for a particular action potential. So we have three waves here. See, these three waves are producing three action potential. This pattern which is produced by this action potential will basically encode for the frequency and go to the cortex. But again, this had a shortcoming. That is, a single neuron, like sometimes the frequency is so high, like till 20,000, one single neuron cannot compensate for so much of frequency, right? So a single neuron cannot take greater than 1000 of frequency. So Wally theory came in. So what Wally theory says is the same thing, that each crest and trough encodes for one action potential, but reception happens by multiple neurons. So let's say the first wave is recognized by the neuron 1, the second wave is recognized by the neuron 2, and the neuron 3 recognizes the third wave. So this is again, this pattern is recognized with the cortex, and the frequency and the pitch is detected. So pitch is ultimately defined or explained by three theories, which is the place theory, the frequency theory, and the Wally theory. The thing is, whenever we're talking about pitch, we are mainly considered where the sound is and what pattern is actually being produced, right? Two very key important points: where the sound is and what pattern are they kind of producing. Talking about loudness, which is basically upon the amplitude of the sound, here we are considered about how many action potentials are produced. So how many, right? In frequency it's where and what, whereas in action potentials how many are produced are concerned with loudness of sound. So this kind of concludes the organ of Corti and some theories of hearing. Lastly, if you were to try to understand the mechanism of hearing, or try to relate to what is mechanism of hearing, understand this simply. Any substance will have molecules within it, and molecules, when are hit with sound, will transmit energy from one molecule to second molecule. In that condition, solids have more molecules, liquids have less molecules, and air has very less molecules per unit volume, right? So sound travels fast in solid, followed by liquid, followed by air. Having said that, talking of mechanism of hearing again, the sound signal which is in the environment is collected by the pinna. It passes through the external auditory canal, strikes the tympanic membrane. Vibrations of the tympanic membrane are transmitted to the stapes footplate through the chain of ossicles, right? And which is coupled to the tympanic membrane. The movement of the stapes footplate causes pressure changes in the labyrinthine fluids, right? And which moves the basilar membrane. I told you, right? The basilar membrane, which stimulates the hair cells of the organ of Corti. And it is these hair cells which are the transducers and convert the mechanical energy into electrical impulse, which travel along the auditory nerve, right? Having said all of this, what is very important is I want you to understand the clinical aspect. The clinical aspect of organ of Corti is important because organ of Corti can be answered in the clinical aspect also. The first thing is we have specific forms of hearing loss. Specific forms of hearing loss as in inflammation of the labyrinth, like viral labyrinthitis, right? What happens in viral labyrinthitis is whenever there is infection through a virus into the bloodstream, there is a tendency of them damaging the organ of Corti. So viral labyrinthitis is the clinical condition. Second is sudden noise or trauma, right? So huge noise or high pitched noise or high sounded noise with high amplitude will cause trauma, which will cause damage to the organ of Corti, which will not let adaptation happen. Having said that, the third important thing is presbycusis. What is presbycusis? It's the sensory hearing loss with physiological aging, right? So presbycusis are usually seen at the age of 65. So what happens here is there is sensory neural loss, right? So this will ultimately lead to the damage of organ of Corti. But remember, presbycusis could be of multiple types. We are specific about sensory presbycusis, which is damage to the organ of Corti. Well, here the condition is like there is damage which starts at the base of the coil and gradually goes to the apex, right? The catch here is that high frequencies are affected, like very high frequencies are affected, but speech discrimination is still good enough. So this kind of ends this complete video. Make sure to